Hello everyone, welcome to this channel. In this video, I'll be talking about fungal staining. What is fungal staining? Fungal staining is an experimental technique used to visualize fungal structures, morphology through microscope. There are two types of fungi are microfungi and macrofungi. Both types of fungi basically grown on moist area, dead living matter or moisture containing foods and spoil them. Macrofungi such as mushroom, wood degrading fungi can be easily seen in naked eyes, whereas microfungi cannot be seen in naked eyes. Purpose of the fungal staining. Generally, the fungi are stained are microfungi and also the spores and body parts of macrofungi to study their morphological characteristics. As most of the microfungi can cause disease in human, animals, birds, insects and plants. It's very necessary to identify their morphology and structures. Based on the microscopic observation, more detailed laboratory investigation is done to understand their basic ultrastructure and the components they produce. So, the structure of fungi. Fungi are eukaryotic, unicellular or multicellular organisms having cosmopolitan distribution in nature. Cell wall made up of chitin and cellulose that distinguish them from other eukaryotes. Fungi composed of thread-like branches of filaments called hyphae, in singular form called hypha, in the group of hypha called Mycellium. So here the structure of Mycellium, and they can be septate or aseptate. In this video here it's showing the septum of hyphae where the cellular organelles are there. The procedure of fungal staining. For this procedure, you need a proper fungal culture, either stored or cultured by serial dilution method from rotten fruits or any other sample on a suitable medium for fungi, such as potato dextrose agar and severed dextrose agar. Then you need to take a clean grease-free glass slide and a loopful culture of the sample plate by using a sterile inoculating loop. Then make a smear by spreading the sample on a clear glass slide and allow it to dry. Then after the place of a drop of lactophenol cotton blue or safranin over the fungal part. It acts as mountain stain which color the fungal cell wall as well as whole elements blue in color. Use a coverslip over the fungal culture in a way that any air bubbles should not be trapped in between slide and coverslip. Now examine under microscope at 40x and 100x. The figure is showing the fungal culture on potato dextrose agar and the microscopic observation of fungi. Here it's showing Aspergillus species, and also zooming the picture of microscopic slide, it's showing the structure of Aspergillus mumps. Composition and mechanism of fungal staining. The stain used to generally is lactophenol cotton blue, and the composition of lactophenol cotton blue is lactic acid which preserves the fungal structure and helps to visualize the structure. 2. Phenol. Here phenol acts as disinfectant. It kills unwanted microorganisms or contaminants. 3. Cotton blue colors the chitin or cellulose of fungal cell wall. 4. Glycerol and distilled water also used to make the stain. Here it's a macrofungi, Basidiomycetes. It's structural that can be visible in naked eye. Applications and advantages of fungal staining. Fungal staining is very useful staining technique used in clinical laboratory as well as research laboratory for the identification and characterization of fungi and the disease they cause to humans and other animals. Based on this clinical diagnosis and research can be done, such as an example of the accidental discovery of penicillium by Sir Alexander Fleming in 1928, that's called world's first antibiotic that saved many lives during World War. Here the structure of penicillium mold a type of multicellular fungi. It's showing here. The conidia, conidiophores and spores are showing here. Here it's also a structure of mold aspergillus. The structure is showing here. The conidia, conidiophore and conidiospores are there, and fruit body also there. And so, thank you for watching. Subscribe for more. Hello everyone, welcome to this channel. In this video, I'll be talking